Dear all, good evening for some and good afternoon for others. Welcome to the 7th week of Ecoastronomy Sri Lanka Lecture Series 2nd edition. Today, dear students, we are both honored and humbled for having with us a special key speaker from Morocco, Dr. Abdelouahed Lagnaoui, Assistant Professor at the Interdisciplinary Research Laboratory in Science, Education and Training, High School of Education and Training, Vertebrate. As well, he is affiliated at Hassan First University of Morocco. Dr. Abdelouahed Lagnaoui, known by his accomplishment and devotion to science and research, he has published and been part of important projects. Ladies and gentlemen, I would love to invite our key speaker to take us in an exciting, wonderful journey through his lecture about vertebrate and invertebrate trace fossils, the unique window into the behavior of extinct animals. Doctor, the floor is yours. You can get started.
Firstly, many thanks for your invitation to give a lecture on invertebrate and vertebrate trace fossils, their importance for earth sciences and particularly for biostratigraphy, paleo.
Doctor, you can open your cam while you're talking, please.
Particularly for biostratigraphy, paleoecology, and paleoenvironmental studies. So, I don't know if you can see the presentation.
Positive, doctor, we can see it.
Yeah, that's fine. So, invertebrate traces, or generally trace fossils, are the unique window into behavior of extant organism or extant animal. It's the only things that give evidence of how animals or how extant behaving throughout several things, behaving in their local community, in their response to the environmental changes and paleoclimatic changes. So, I would like to mention that ethnology is the science of trace fossils, and some students mix it with ichthyology, which is the study of fishes. So we will talk only on ethnology, which is the study of trace fossils, not fishes. So I will give a brief introduction on ethnology. Paleoethnology is dealing with study of fossilized footprints, tracks, burrows, and other traces as evidence of the activities of organisms. So, as you know, this is the study of ancient trace left by organisms, while neoethnology is the study of the activities of the living animals. Generally, ethnology is the study of trace fossils, while ethology is the scientific study of animal behavior, usually with a focus on behavior under natural conditions and viewing behavior as an evolutionarily adaptive trait. So it's a part of ethnology, and ethology it's linked with ethnology or the interpretation of trace fossils. So to understand any trace fossils, we have to rank or class the trace fossils. So we have the Econo Taxonomy. It's the parallel synonym of taxonomy used for the body fossils or skeletal fossils. So we use Econo Taxonomy. They have the same code as the International Zoological Code. We use the same binomial names, genus and species. So, but we add Ecno, and we discuss about Ecno genus and Ecno species. They have the same rules as the International Code of Zoological Nomenclatures. It's an artificial, of course, classification of trace fossils. So then, after this, a little bit introduction to diverse terms used in ethnology, I will give a little bit some history or marked steps in the history of technology. It's an ancient science dated more than 1830s when early geologists described dinosaur tracks as giant animals. They found that most of the important scientists who discussed technology, it was Leonardo da Vinci, and he is called the grandfather of technology. He described several invertebrate traces in his notebooks without giving any interpretation or other things. So he considered these trace fossils as annihilation. Others and he discussed some fossils like worms in this notebook without knowing that this is not worms, but this is invertebrate traces of different type of organism. So later, or when Edward Hitchcock came and described the first dinosaur footprint, but he described that as a giant birds, and called it the first time ichnids, and called it the science as echnolithology. But later, some scientists reduced the name to echnology and changed ichnids to different echnofossils, trace fossils, and used some suffix and prefix in the name of this echnotaxa or this trace fossils, mostly finishing by ichnus or ichnitis and so on. And this was the first publication on vertebrate tracks or dinosaur track without knowing this is dinosaurs before the discovering the skeletal of dinosaurs. So it was the first description of vertebrate tracks, dinosaur footprints, as giant birds tracks. It is only in the middle of 20th century that the study of trace fossils became more important with the introduction of its importance in the paleoenvironmental and the paleoecological analysis by the German geologist Adolf Seeliger. So he make a classification of invertebrate traces and give an important book on the use of invertebrate and vertebrate traces in paleoenvironmental interpretation in paleoecology and paleoecosystem setting analysis. He was called as a father of the modern ethnology. And He published diverse books on ecology, classification, description of their importance, and some examples from diverse area. He suggests first time 5 main behavioral classes, which are Capecnia, means recent traces, Domicnia, which is dwelling, Phodicnia, deposit feeding and mining, Passicnia, and Ripicnia. This is the main 5 behavioral classes suggested by Adolf Selecher. Later, in the rise of ecological studies, there are a lot of books by Richard Bromley, Ben Burton, and Louis Botois, Dick Knoss, and several books who start to make the point on the importance of trace fossils as an indicator of sediment environment, use it in paleobiology, taxonomy, and other use ecology in the exploration of petroleum studies. So after that, after 1970, the ecological study rise in a great increase in fastly, and we have a lot of books describing the importance of ecology in different topics like paleoenvironmental stratigraphic analysis, petroleum exploration, and so on. So now, to understand the trace fossils, even invertebrate or vertebrate, we have to see how we have to see the interest of this trace fossils. So let's describe them as a biogenic structure. Trace fossils, they are a biogenic structure. Even zoogenetic structures or phytogenetic structures, they have a wide stratigraphic distribution from Precambrian to the nowadays. They can make a relationship between trace or tracks and the producers who make this trace, and can give information on the date of this animal. It is characteristic by typical structures. Each organism produces its trace with a typical way, so it's typical structures for its group of animals or group of organisms. And mostly we don't know the producers. Only in rare cases we can find the trace or the footprints with fossilized with the producers of the animal who produce this trace or these tracks. So mostly we don't know the producer of these trace fossils. So I will give some examples on these traces. For example, here we can see a lemur fossilized with the trackways. When this lemur walk, and at the end of this trackways, it did. So we have the animal and its trace fossils, and this is also, as you know, when we work on geology, we use the principle of actualism. So we correlate what happened now with what we have in the past. So this behavior is recorded in the current coast in the North America, and we can see the same structures in the south of Asia. So it gives an information about the date of the animal. And we have the second, we have some typical structures. For example, when we see a trace with three digits, or it gives automatically or quickly an overview that this is produced by the dinosaurs or any one of the group of dinosaurs. We have also the typical structures of sauropods and ornithopods. Archosaurs also have a typical structures with a pentadactyl footprints and pies and menus, which are, if we can say, hand and legs, footprint of hand and legs, pies and menus, imprints, and it is characterized by 5-digit imprints. Rarely we can find an animal or an organism with its trace, so only in rare cases like the fossilization of soft bodies organisms in their burrows. Some examples of polychaetes, annihilates preserved inside the trace. We have also some burrows with shells of gastropods, of bivalves preserved inside their trace. So, ethnology, as I say, it's the study of trace fossils of animals and plants, generally including plants also. It includes the study of the behavior, which means the adaptation of the animals to the environmental settings. And this is give also trace fossils are used as indicators of the environmental and paleoecological conditions during the sedimentation. So when we discuss neoecology, we are dealing with recent traces, and this is include biologists, ecologists, and oceanologists. And this is the principle of uniformitarianism or actualism used for correlation with the fossil, with trace fossils, which involve paleontologists, sedimentologists, and sometimes geochemists. So there are a lot of a lot of authors try to understand the biogenetic structures and then the trace fossils. They are considered as biogenetic structures. A lot of activities, including bioerosion structures like perforation, craters, and also tools used by human. Homo sapiens also is considered as trace fossils, eggshells. And then we have the biogenetic sedimentary structures. When we have the interaction between the organism and the substrate or sediment. So we have done the different bioturbation, including tracks, cast, tries, burrow, grazing, boring. So a lot of kind of trace fossils. Different from the biosedimentation, which is mainly related to fecal pellets or fecal casting. Then we have the biostratification structures or bioconstruction, when we have kind of deposit building by organisms, including recipal bioconstructions. So paleontologists and paleoecologists try to understand the major step of life on Earth, because we have the, as we say, the trace fossils are recorded from the Precambrian, and during the Cambrian explosion, we can also record the oldest record of vertebrate footprint, which mark the transition from the marine life, and we have the amphibians, this transition of amphibians from the marine life to continental life or terrestrial life. So this is one of the most the interest of the study of trace fossils. So it can be a piece of evidence of the major steps in the history of life on Earth. So we have, from the point of view of behavior, we have different kind of behavior, and we will discuss this behavior in some details. So, and this behavior of organism recorded in deposit or in sediment, it's a kind of response to different environmental conditions. When we have a change in the environmental conditions, including if we have increase of the water table or decrease of water table, the organism responds to this change in the environmental conditions, and then this response is recorded by a typical trace fossils. So, and this also include the food case, searching for food, protection against predators, protection against current, against the current, against the rise of sedimentation rate, against the heat, against the desiccation. All this behavior, it's a response to environmental and ecological settings. So, and we have divers that we will discuss in detail. Each one like feeding, dwelling, migration, racing, locomotion, and so on. So this is in the same way. When we have a change in the sedimentological condition, we have also a response of this organism to this change by changing their behavior, and we have typical traces responding to this sedimentological change. So, and this is include changes in bathymetry, in salinity, anoxy, agitation, high energy or low energy. We have a stop of sedimentation, decreasing or increasing of the sedimentation rate, diagenesis, substrate lithification or compaction, and all this change in sedimentological condition are recorded by trace fossils when we have, of course, trace fossils. And then we have the notion of ecnofishes, which correspond to the association of ecnofossils with the typical Econo taxon or index ecnotaxon, which indicate a particular condition during the deposition. So, one of further interest of trace fossils is biostratigraphic correlation, and we have some trace fossils are typical of an age, typical age. If we take an example of Arenocolitis franconicus, it's typical recorded from the Middle Triassic. Generally, it firstly described from the Germany. And we have also some typical forms of Eocene bird tracks from Eocene. We have also other invertebrate traces with the hexagonal structure, and this is recorded from Silurian to Tertiary. We have Odohamia from the Cambrian, Crisiana from the Cambrian to Silurian, and so on. Also, it can give an indication and use it in the Cambrian to understand the Cambrian explosion. And we have diverse record of trace fossils, and each typical for time. If we take, for example, Rosophycus avalonicensis is typical from early lower Cambrian, and so on. And this is recorded in the same way as trilobite, and mostly this kind of traces are produced by trilobite. Recently, a study correlates diverse Cambrian trace fossils and used them for a biostratigraphical and trans-regional correlation between Jordan, Israel, Egypt, Libya, Tunisia, Algeria, and Morocco, and give a good indication for this for the assignment of the age of some formations when we have lake of fossils or body fossils. And I will take some example from Morocco as well. We have the Ecnogenous Protocheirotorium, which is described from the Ergana Basin for the first time in 2010. And this is the first time where early Triassic described in Morocco. Before the 2010, the Triassic deposits are known only by middle and upper Triassic deposit. Never early Triassic described for Morocco, and throughout the discovery of tetrapod footprint of Protocheirotorium, now we have the early Triassic in Morocco, and we have here some biostratigraphical distribution of different tetrapod footprints, including Ecnotaxa from the Carboniferous to Permian and others from the Triassic and early Jurassic. All these Ecnotaxa, there are some Ecnotaxa who give a good indication on the age of the deposits. If we take, for example, Cheirotorium barti, which is typical for Ladinian, Anisian Ladinian. If we take, for example, Protocheirotorium or Apatopos, which is typical for Carnian, Norian, and so on. So, to understand ecology, I will try to summarize the different principles of ecology. So, firstly, to understand ecology, we have to understand that a single animal or a single organism can produce several types of traces, recording several types of behavior, and we will take the example of some some mammals, like here we have a Gnaeau marks, we have fishes, we have footprints, tetrapod footprints, and we have burrows, like a complex network burrows. So this is the only a single organism that give us at least 5 types of traces, and then 5 types of behavior. So we can say that an organism can produce dwelling burrows, feeding traces, locomotion, fecal traces, and escape traces in case, then representing 5 different behavior. If we don't know a little bit of the, if we don't know some, how I say, ethological studies of recent animals, we can misinterpret these traces as produced by different organisms, not only one or single. So we can see that these different traces of a point of ethnologists after a correlation between diverse structures, then we can realize it was produced by a single organism. Second thing, invertebrate or vertebrate trace fossils are recorded in sediment. So if we have different sediment, for example, we have fine-grained sandstone with coarse-grained sandstone or with klystone, we have a different diverse type of preservation. So we will get different results, and then the only or single traces can be preserved in different ways according to the nature and the consistency of the sediment. So we have to take in consideration the sedimentological settings and the taphonomic conditions to understand and interpret trace fossils. So second one or third principle is that, for example, if we have two animals with the same morphology, we have two animals with the same morphology, these two animals can produce similar traces, and we will take examples. In the Cambrian, we have trilobites. They are arthropods, and we have similar traces in morphology from the actual some arthropods that they have similar traces of trilobites. Then these two group of animals can produce similar traces, and if we don't know this, we can misinterpret this about traces as produced by the same animals, and this is not real, because when we found some type of Cruziana and Rosophocis from the Cambrian, we can say these are produced by trilobites arthropods. But when we have the same traces, I mean Cruziana and Rosophocis from the Triassic or the Jurassic, we don't have any more trilobites after the Paleozoic. So what kind of organism can produce Cruziana and Rosophocis? They should be organisms with the same morphology as trilobites, I mean bilaterally symmetrical organisms. So, and we have diverse concostracans, polychaetes, worms, or snails that can produce the same traces, I mean Rosophocis or Cruziana. So this is also to understand and interpret invertebrate traces, we have to know the biota of each ages. For example, when we discuss the Cruziana and Rosophocis in Triassic or Mesozoic, it's not the same when we discuss Cruziana and Rosophocis from the Paleozoic. So we have to know the biota of each age and try to make an interpretation in the light of the fossil finding or of paleontological record during that time. So furthermore, that make ecology complex is that the trace fossils is range from sample, and this is easy to find sample trace fossils. and too complex, and the complex, this make a little bit difficult to interpret trace fossils. These complex trace fossils or could be recorded as network, as you see here below, a network of thalassinoides, which is a complex network that could be produced by one individual or different individuals, and could be also by one group of animals or more than one group of animals, and that make a little bit difficult to make an interpretation of the complex trace fossils. I see in both marine and continental settings, we can find this type of complex structures, which are mainly composed of structures or gallery of different size and shape. The preservation of trace fossil also given an important indication on the fossils, on the trace fossils and its classification. So we have mainly 5 groups of trace fossils according to the preservational conditions, and we have even epirelief, convex or concave, or we have the full relief or hyporelief, convex or concave. So this kind of preservation also can give indication on the type of behavior and then the classification of trace fossils. So same as the complex, we have also simple component or composite structures, and this kind of preservation exists in different record of trace fossils in the world. We have sample preservation, which is the most typical of trace fossils. It's an isolated trace fossils indicate one behavior produced by one organism, and we have a component trace fossils represent interconnected trace fossil morphologies produced either by different organisms or by the same organism with different behavior. And we have a composite trace fossils represent one trace fossil overparenting or constructed within a previous existing trace fossils. When we have superposition of two trace fossils, we can then discuss about composite trace fossils. So I will go quickly in this part because we don't know how much we still have time. Anyway, so most important.
We still have time.
We still have time. Great.
Yeah.
Because I still have a lot of slides to show lot of things about technology, but
keep it as as you like to do it, just like
okay.
few informations. Thank you.
Okay, thank you. So the most of trace fossils are produced by soft bodies organisms. So what we can say about this, that organism, when we found one organism, we found only one fossil record, and this fossil or this body fossils or organism recorded as fossil can produce a lot of trace fossils. So the biodiversity recorded, I mean that the biodiversity recorded by trace fossils, it's much more abundant than the body fossil. So most of these organisms could be also vertebrate plants, fungi, protists, or bacteria. And the record or the preservation of this fossil, it depends of the environmental settings and the taxonomic condition during the deposition. So even in a marine or continental setting, we have the distribution and abundance of trace fossils that can give information on depositional energy, sedimentation rate, bottom water oxygenation, a lot of information related to ecology during the time of deposition, even in the continental or in the marine deposit. And we have here a study on the Jurassic deposit from Algeria. We can base it on invertebrate Ecno assemblages to see the distribution of this Ecnotaxa and what kind of information that give on the storm sequences, on granulometry, echnofabrics, bathymetry, hydrodynamics, energy, oxygenation, nutrient, and other information related to sedimentation rate. So mostly the invertebrate traces have a wide vertical stratigraphical distribution. So it's rarely used in biostratigraphy. I mean, most of invertebrate traces. But when we discuss about tetrapod footprints, they are very useful for biostratigraphy when body fossils are missing. This is one of the interests we discussed of trace fossils, mainly vertebrate tracks. Same thing, here we can see a lot of conditions and interaction between substrate and the organism that live footprint trackwise or any kind of trace fossils. And all this kind of conditions, even in sedimentological conditions or soil conditions, water table, oxygenation, presence or absence of organic matters, and so on. All these could be recorded and indicated by the Ecno assemblages recorded in the sediment. So trace fossils are strongly controlled by groundwater profiles or water table, by the sedimentary fishes, and so on, even in the marine or continental settings, I mean. So this is like I guess this is like the same thing. They changed the. So this is as I say, a lot of conditions, ecological conditions, environmental setting is recorded or indicated by the assemblage of trace fossils recorded in the sediment. So we can discuss sedimentation rate, depositional energy, media consistency, groundwater profile, water viability, oxygen, carbon dioxide levels in soil, atmosphere as well, in groundwater, temperature also, So this is mainly the main use of invertebrate traces and as indicator of the environment and ecosystem. So trace fossils are recorded in situ, which means in the sediment. It's not like body fossils. And this is briefly, we can say that trace fossils are typically more abundant than body fossils in most sedimentary sections. So this is the. So now we have to discuss how we can, what kind of discipline that can use to understand trace fossils. There are three main parameters that control the study of fossils or control trace fossils. We have the behavior, we have the producer of or the animal or organism, and we have the substrate, the sediment. So, and each one of these three elements can make an influence on the other elements, and then we have some relationship between the producers and the behavior, which give an ethological insight. And we have also body implant, we have producer in interaction with the substrate, and we have the substrate with the behavior. It give also indication on sediment mechanics. So these three elements, key elements to understand the trace fossils. We have discussed that we have diverse, of course, classification of biogenic structure, which is what is the most important is the biogenic sedimentary structures. Others are important. We have biostratification structures like stromatolites. We have bioturbation structures like tracks, trial, and burrows, and biodeposition structures like fecal pellets, pseudofishes, and coprolites. To make a classification, it seems that we discussed it in the introduction. We use the International Zoological International Code of Zoological Nomenclature with binomial type, like we have genus and species, and for example, but we call that ecnogenus and ecnospecies. Ecology, we have other groups like other rank like classes, families, but they are not much used in ecology. So what is the most problem to understand ecology is to find the organism who made these trace fossils. Only in rare case we have an association of the trace fossils and the body fossils. Sometimes there are some confusions used in ecology and biology. We have the ecnofossils or the trace fossils. It's called Arenocolitis, which is not linked to Arenicola, the organism. They are not produced by Arenicola, but this kind of nomenclature gives some confusions, and some non-specialists of ecology could make a link between Arenicola and Arenocolitis. It's the same between the polychaete nereis and the ecnofossil neretis. So they are not produced by nereis. Nereids are not produced by nereis, and there are some confusions. This is two examples, but there are a lot of examples also. So we have to see that the same species can produce different type throughout the same behavior in different substrate. If we have a soft substrate or hard substrate, if we have a wet or dry substrate. So this is the main issue in ecology. Also the activity when we have the different behavior from the same animals. So here we can give an example. For example, we have the same animals can produce different traces in the same substrate. For example, we have Scalerotuba, Neuritis, Neoneuritis, and here the kind of preservation also it's influence the type of trace fossils. And we have, if we take the example of some crabs, we have this kind or group of animals can produce different traces throughout diverse behavior. And we have, for example, Psilonicnus, which is a G-form structure. Number one, we have Diplicnids, like a trackway when the organism walk. And we have some grazing traces and fecal balls preservation. And this is the four behavior are produced by the same organism. We can take also the example of trilobites, and in trilobites we have at least 3 known behavior which produce 3 traces. We have Rosophocis when the trilobites or the organism is in resting position, and we have Diplicnids when the organism walk. It's a kind of locomotion, and we have Crisiana. It's a kind of phodicnia when the organism looking for food while walking. So here the same organism gave at least 3 types of trace fossils and then 3 types of behavior. So some we explain some kind of traces produced by different organisms. So if we take, for example, Thalassinioides, it can be produced by crustacean, can be produced by some fishes, crabs, and so on. Also, Rosophocis can be produced by polychaete worms, snails, shrimp, and trilobites. So, to make a classification and based on previous studies, we have to take in consideration the interstratum structures, including shape, branched and branched, structure of the filin, structured or homogeneous, size, arrangement, horizontal, vertical, inclined, random, and we have to see in the wall morphology. Also, we have to analyze the surface structures, and we use these 5 parameters. And we have, I don't know if you see here, we have like crescentic ship filling, we have Y branched structure, we have a meniscate structure, and temple and branched traces. So we see already this classification of the kind of preservation of trace fossils. So we can go, there are 2 classifications. Selecher suggests 4 relief, semi-relief, epirelief or aporelief, while Martinson suggested exychnia, epicnia, andychnia, and so on. And then this is the most important thing, it's the ethological classification based on behavior of animal. And we have 7 main group of animal. We have Cabechnia, which means recent trace, Repicnia, locomotion, and we will see this slide for each one. And each kind of group indicate typical behavior. Here are the most important kind of behavior or ethological classification suggested by Boutwa and Mongo 2011. So we can start first by the recent trace or Cabechnia. Recent trace or Cabechnia, it's produced by organisms when they are in a recent position. And then we have, for example, trace like a star for starfishes, which is called Astreatis, and we have rosophocis for arthropods. And this is give a typical structure of the ventral part, or give more detail of the ventral part of the organism. We have several examples, including Lochia for bivalve and Selenignits for Lemurés and Striatics, Rosophocis, and so on. So, which is the most important? That Cabechnia indicate a resting position of the organism and give more detail on the ventral anatomy of the organism. Locomotion or Repicnia, it's a trace produced when the organism walking, moving from a place to another. It's mainly known by like dinosaur footprints, dinosaur trackways, reptile, amphibians, and some arthropods can produce diplinic nets for example, trilobites and arthropods. Cophecnicium or Paleoelcura also is a part of this group of traces. Passechnia or grazing trace is the structure with the combination of locomotion and feeding, looking for food. When organism walking or moving while searching for food, this is called passechnia or grazing traces. It's mainly recorded in mobile ephonal deposit feeders or epiphonal detritus feeders. It's mainly a sample traces that reflect locomotion typical for each type of organism. And there are a lot of groups like Gordia, Myrmia, Psamicnids, Nerids, and so on, including even Squinia and other groups. And this group, it's mainly membrane-shaped sample and horizontal. So we can take some example of Rosophocis, which have diverse morphotypes with different shape and morphology, and each one is typical and indicate how this organism responds to the environmental and sedimentation changes during the deposition. Farming traces or agrichnia, it's like that produced mainly as a perfect geometrical structures. For example, we have hexagonal structures, we have other groups called mainly grabtolids. This kind of group, or this group, it's combined dwelling and feeding activities, and mostly interpreted as bacterial farming or traps produced by some meiofauna or microorganisms to searching or looking for food, or even for other activities related to bacterial farming. It's mostly regular burrows pattern with the typical geometrical forms, morphologies. And this include Paleodecton and different Proto-Paleodecton, Megagrapton, and so on. They are known as Grabtolidis. And here we can see the structure, how it looks inside the sediment. And sometimes we can see only this form with the systematical points in the bedding plane. Then we have feeding trace or phodicnia, which is a behavior dealing with the dwelling and feeding activities. It's dwelling and looking for food, typically produced by ephonal deposit feeders involving underground mining strategies. They have a wide range of morphologies with sometimes simple to complex structures. Common patterns including simple burrows, sometimes branched like chondrites or ficodes, sometimes with U-shaped structures like rhizocorallium and so on. Dwelling traces or domicnia is a permanent habitat of the organism, mainly produced by ephonal organisms. It's a kind of domicile or permanent domicile of organism. It's mainly not record on one behavior, but could be more than one behavior in the same structures. The morphology of these traces is sometimes with the vertical tubes, including U-shaped tubes or branched burrows, like Arenocolitis or Diplocritarians and so on. And when we have also discussed this kind of groups, domicnia, I mean, and why the organism produce or construct vertical tubes. And this is only indicate that the environmental settings is characterized by high energy, because the vertical structures could be a kind of the protection versus the current or the high energy, high hydrodynamic energy. So the vertical construction indicate that kind of high energy, while the horizontal trace fossils indicate a stable substrate with low hydrodynamic energy. Also, we have the phodicnia or escape traces, and this is mainly recorded when we have a change in the sedimentation or ecological conditions. So when we have this change in the sedimentation or ecological condition, the organism try to escape and stay in what we can say, look for the better sedimentological condition. And mainly it's associated with the rapid or quick sedimentation rates. So we have some examples from the Argentina when a type of bivalves, elongated bivalves recorded and assigned oblonignus and indicate a kind of escape from when the sedimentation rate became more, increased more and more. We have also equilibrium traces, and this is when the organism look for better conditions to keep or to equilibrate the conditions. I mean the ecological and environmental conditions. When we have any change, this organism try to leave the, what we can say, leave the gallery or the tube and try to look for the equilibrium situation in the sediments. And this is also indicate that there is a progressive aggradation or degradation, reflecting gradual adjustment under background sedimentation. It's mostly recorded in euphonal organisms, which move up or down prior to maintain certain depth with respect to sediment, water, and surface. Mainly, it's recorded in the Crocodiles, and we see here the Crocodiles. It's the vertical eutipid with Brighton and the Rhizocorallium or Roselia or other vertical invertebrate traces. We have also more technia or the traces. It's when the organism, how I can say that, it indicates the last activity of the organism before death. So this is called mortechnia, and give sometimes, and like in other some region, indicate even the cause of the date when we have indication, sometimes some recorded in Germany with when this kind of fossils associated with the volcanic ashes. Sometimes this kind of trace, most important to get from this, sometimes it gives the cause of the death, and it considered as the last activity of the organism before the death. We have also predation traces or peridychnia, and this is when we have two organisms in competition, or we have predator or prey, and someone leave traces in the other organism during this competition. This kind of traces, predation traces, is recorded mainly and most abundant in dinosaur bones. When we have bones with some teeth marks, it could be assigned to the same group, which is predachnia. I guess this is the last group of the classification is nesting trace or calcicnia, and it's mainly composed of nest or excavated area for breeding purpose, mainly produced by insect larvae, and give some kind of structures like cells and chambers linked with some galleries or tubes. And we have sometimes sometimes we have combination of one or several behavior like dwellings, feeding, recorded by ophiomorpha, and or dwelling or thalassinoids as well, which has vertical burrows, network burrows with diverse structures. Here are the same scoletos also can be produced by combination of dwelling and feeding, arenocolitis, U-shaped structures, the same things. So now I will try to give quickly some exceptional recorded behavior in the world, and I will start by the ancient one, the most oldest evidence of activities. From Franceville Bassin in Gabon, a group of researchers recorded the ancient life, evidence of life, which is, I guess, 2G years. And this is a kind of bacterial structure. But recently, the same group recorded a type of burrows. And these burrows, in a primitive life, give an insight that we have several movements. We have vertical, horizontal, and inclined movement. And it seems that this is really a complex movement, and not only the primitive organism could produce that. They should have some kind of structures like annelids. It's a questionable traces recorded from this, and most important that indicate that the organism during that time recorded during the great oxidation events could move in all direction and could construct the galleries also in vertical position, in horizontal and inclined position. And anatomically, to produce this kind of structures, this should be more developed than the researcher thought. It's not so. It's not a primitive kind of life, but should we have any kind of structures that allow movement, changing the direction or changing the position. So it's a little bit complicated to understand this invertebrate records from the Precambrian. So also some traces could be mixed, and we have here similar combined Diplocoretarian, Arenocolitis, Scoletos, and this behavior is mainly recorded in crabs when the organism is introduced in the gallery from one aperture and try to get out from the second one. And this type of spriton structures linked bow tubes, it's mainly a kind of behavior for searching food and trying to stabilize the structure of this gallery. So what I say is that anatomical studies or body fossil studies give only some insight on the anatomy and morphology of the animals, of the organism. But to understand the behavior of this kind of organism, and like fish, for example, we have to record their trace, and mainly the fishes are produce trace belonging to Odichnia. And recently, an Odichnia was described from the late Devonian of south of Morocco, and this is indicates a kind of behavioral swimming traces, like in sinusoidal movement of the fishes, and give some insight of how the dynamic of these fishes or trace makers happen. For example, we have the vines, pelvic vines, and how the body moves. If we take, for example, the yellow part of the body, it's a little bit semi-static when the animal doesn't move this part a lot. The part of the body that moves mostly is the green one, which is the most part moving, and leave these main traces in the middle in the green traces. Also in continental seeding, a lot of echinotaxa and diversity, echnodiversity include record diverse behavior in the continental deposits. And we can take the example of Paleontologist described from Morocco. And if we take the normal and the before this time, the main recorded scorpion traces include three traces with midline, while this type of traces described for Morocco, it consists of 4 legs with 2 marginal traces and sometimes median traces. This is produced by scorpion group, and I will give an insight how we can make an echnotaxonomical analysis, or how we can distinguish echnogenera and echnospecies. I will give an example here. Firstly, we can find or discover like these traces. We have to see all recorded traces, and here we can see that we have 4 traces with median traces and 2 marginal structures or drags. This traces is more than 1 meter long and give an insight how the animal or the scorpion move and make a turn in the right and so on. We see the structure of these traces and make like a statistical analysis of the position of these 4 traces. We compare them with diverse recorded structures. We have here Arenocolitis dunbari and Arenocolitis tridactylum with 3 traces recorded. We make a correlation of the position of these tracks according to the midline, and then we can see where is the difference between this kind of traces. And we can also make a correlation in the position of the midline, the arrangement of the 3 traces, and so on. This arrangement can, for example, here the angle of this alignment of these 3 traces with the midline, it's larger than the arrangement of these 3 traces with the midline of the Paleontologist. Here we have the difference in the arrangement of this kind of traces, and we have the morphological and patterns of these trackways, and that make it as an exceptional structure produced by scorpion. And normally scorpions traces are known by Paleoecula tridactyla and Paleoecula dirbae, and recently this discovered one, it's Paleoecula from Morocco, given a new behavior produced by scorpions. And this is by the current observation of scorpion behavior. When we have scorpions mainly walk on three of their legs, but when we have a large one, they walk on four. And sometimes when some scorpions are pregnant, they walk on four legs, different from the other scorpions walk on three legs in most of the time. So this behavior also can give this information on how this scorpion walk and why here four legs recorded, but in the other behavior only three legs recorded. Also, the external structures of some invertebrate traces can also give indication on the behavior, why some invertebrate traces make back filling, others more transversal striation or crescentic striation. And this is also recorded by different behavior from species to another, or from ecnogenus to another. So also for vertebrate ecology, we have a bipedality record of some crocodiles from the Cretaceous of South Korea, and this is the only crocodile who walk on two legs. That means it's bipedal animals, not like the current or the actual crocodile. This behavior, it's interpreted as related the anatomy of this crocodile, but could be also a permanent behavior recorded in this group of animals walking or trying to escape some predators or following some prize. Most epic behavior recorded in a group of theropods from the Cretaceous, it's that some groups of dinosaurs have ceremonies before meeting, before make some sexual relation, and the dancing or singing or some kind of this behavior. The recorded footprints from North America indicate that this kind of movement of dinosaurs, it's like dancing. We can see movement in all directions, like dancing or like a party ceremonies before meeting and make a relationship. And this kind of behavior is dancing and singing is recorded in some birds from the current observation. And this is also one of the epic description published in 2016 on this kind of behavior recorded from Cretaceous deposit of North America. Also from South Korea, a lizard tried to escape from a predator, and to increase the vitesse and increased the speed, he changed its behavior and start to walking on two legs, change from quadrupedality to bipedality to increase the speed, trying to escape from the predators. And this is also recorded from the the Cretaceous deposit of South Korea. So here I can give a short, quick how we can make a distinction. For example, we can trout invertebrate, trout tetrapod footprint track-wise. We can make a difference between amphibians and reptiles because each one walk on with a different way. And this is related the posture of the body. We can also make a measurement to see the body size, the body length. Also, this kind of trackways or trace left by dinosaur footprints can also give information on the structure of the body of the animals, and we can see when we have narrow trackways, it's not the same when we have a large distance between marginal borders of the trackways. And we can see that in both type of trackways can give information on the structure of body. And if we have a like a posture, elongated or elongated legs or erected legs, like the second one, A, it's more erected body posture, and the second, it's not erected, it's more large posture. This is a study from Argentina on these cyanodont groups live in trace fossils, and this is could also give information on how the position of the body of the animal and structure of the body of animal can also give indication on the structure of the feet and the size of digits, posture, and if they have palm or not, and the structure of the also of the I see the length of the body and distance between both the posterior and anterior legs. And then we can have a restriction of the animal and how the animal can walk with this kind of primitive cyanodont group. Here a representation of this kind of studies can give an important information on the structure of legs, how they are arranged, the posture, how it's arranged, and so can give also how the animal can walk. We can also calculate the speed of walking. We can see the size, the height of the animal. All this we have a formula from we can calculate it from the trackways and the footprint left by the animal. From Germany also kind of behavior recorded in archosaurs, and we have here give different preservation of the animals when he walk in soft substrate and when he walk in hard substrate, and when he changed the behavior and start swimming, give different traces, and when he found some cry how they can behave, and all this kind of feeding behavior is recorded from the Triassic, middle Triassic of Germany, central of Germany, produced by archosaurs. And we can see in here are the drawing of the tracks with the lemurs, which are the main food of the archosaurs in the coastal area. So, as you can see, we have several formula to calculate the size of the animal, to see how the animal works and the behavior of animals. We can use also some new technological techniques like photogrammetry, lidars, or radars, or some kind of this new techniques to record this trace fossil and make measurement and statistical analysis. So this is a work. It's in progress. It's recorded from Morocco, and we have trilobites or two behavior recorded in the same area. For example, if we take this is considered as Rosophocus, but this is considered as diplonicnates. The trilobites have some stops. This is a resting traces. Rosophocus is a resting trace. This is a stop. The animals stop the movement, stop the activity, then start to walk till this position and stop again. Then start to walk and stop again. And this kind of behavior indicate what? Even this animal is escaping a predator or try to hide itself from the predator by stopping the time from time at the time, and or a kind of looking for food or try to make a traps for some other organism which are the main food of trilobites. Here is with more details this moment of stop of this animal, then it start to walk and produce diplonicnates, stop and produce Rosophocus. Of course, it's this behavior or mixed behavior of resting trace and locomotion is recorded in several plates for several meters. We have 3 plates, the first one and second one. This one 3 plates with this movement, and this is for a long distance, which is more than 4 meters, or this kind of behavior is repeated. And we have this kind of traces. We have diplonicnates recorded in the same time with Rosophocus. So this kind interpreted, as I say, this stops between a moment of walking could be even to escape from the predator or to look for food or take a kind of rest for looking for food or make a traps for some nutrients. So, and this is mainly recorded in this time, and the structure indicates the sense of movement, where the animal is moving, the direction where it is moving, and so on. So this is the last. I speak quickly because it was too long to give too much information in short time. So that's all. Thank you very much. And if you have any kind of discussion or question, you are welcome.